All right, we're going to take a look at the cell cycle. I'm sure you're already pretty familiar with at least mitosis, right? Because you've got the prophase, metaphase, anaphase, telophase. But the cell cycle is actually includes all of interphase and mitosis. It's the cycle that cells will go through, at least cells that have the fate to divide. But the part of interphase, which is not part of mitosis, is broken up into three other parts. So there's actually four parts to the cell cycle. So there's G1, S, G2, and then finally the M phase, which that involves mitosis and cytokinesis. Let's take a look at these in more detail. All right, so during all three of the interphase phases, G1, S, and G2, so the G stand for gap1, the S is like DNA synthesis, and G2 is gap number 2. But in all three of these phases, the cell is doing all its normal activities that it would be doing, but it's going to be growing by producing proteins and cytoplasmic organelles, like it's going to be copying organelles and making more of them, like the mitochondria and the endoplasmic reticulum. But there's some things that happen, this S phase itself, this is where actually DNA replication occurs. So in DNA replication, you've got 46 chromosomes, but each one of those chromosomes are going to replicate themselves. There'll still be 46 chromosomes, but now they're going to be made up of 2 sister chromatids. We'll look a little bit more at that in a little bit more detail in another screencast. All right, so the S phase is the synthesis of DNA, so DNA replication occurs here. And then this G2 phase is essentially what's going on here that's different than the other phases, is just that the chromosomes are actually going to start to condense. And we'll take a look at what chromosome condensing looks like here in the next slide. And then lastly, the M phase is made up of mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. All right, so you need to be able to outline these phases. What's going on in all of them is essentially growth, and it's carrying out cellular respiration, it's carrying out protein synthesis, and all those phases. But in these interphase phases, there's going to be a lot of cytoplasmic organelles that are going to be kind of reproducing along the way. And then we get the special S phase for synthesis, and then we start our condensing of chromosomes here. Okay, so let's just take a look at chromosome condensing that starts to happen around the end of the G2 phase. And your chromosomes are actually really long and spindly, and they aren't. So what we see generally for chromosomes, like they look like these X's, right? This is what you see when you see a cartoon of a chromosome. Well, that's what they look like when they're just about to divide in mitosis. But normally they're long and spindly, and they're about in this part. They're just spread out. You can't even see them in a microscope because they're only about 10 nanometers in width. And if you look at the difference between interphase and, when mitosis starts, it's this long, spindly, the chromosomes are long and spindly. We call it chromatin at this point when it hasn't condensed. So the way that chromosome condenses is because it coils itself in, human and animal DNA, eukaryotic DNA, essentially, even plants and fungi. There are these proteins and they're called histones, and the DNA wraps around those histones. And when the DNA wraps around those histones, that's called a nucleosome. All right. And these nucleosomes allow it to pack in super tight, and then they fold back and forth across each other, and then they create these cool loops back and forth, and then that folds back and forth even further with a protein scaffold until you finally get this thickly condensed chromosome. This won't appear until actually when you're well into prophase, part of mitosis, but this packing begins during the end of the G2 phase.